Good morning, everyone, and welcome to the last DNA Dialogue hosted by Australian Genomics. Before we start, as we come together today in the spirit of reconciliation, I acknowledge the traditional custodians of the country throughout Australia and their connections to land, sea, and community. I'm joining you today from the lands of the Wurundjeri people of the East Kulin Nation, and I pay my respects to elders past and present and extend my deep respect to all Aboriginal and Torres Strait Islander peoples joining us for this webinar today. During the webinar, please ask any questions using the Q&A function that you can see if you use your arrow at the bottom of your screen. And if you're active on social media, we encourage you to use the hashtag DNA Dialogue and at OzGenomics. So we're coming full circle in today's DNA Dialogue after over 27 virtual seminars. The first of these was with Heidi Rehm, who is without doubt one of the leaders in genomics internationally. And we're ending with her today, which I think is incredibly fitting. Heidi Rehm is an investigator in the Centre for Genomic Medicine at Mass General Hospital. She's co-director of the program in medical and population genetics at the Broad Institute, as well as chief medical officer at the Broad Clinical Laboratories. You would know her as the principal investigator of ClinGen and Nomad that are providing resources to support the interpretation of genes and variants. Heidi co-leads the Broad Centre for Mendelian Genomics and Matchmaker Exchange, which is focused on discovering causes of rare disease. I've worked with Heidi side by side in the Global Alliance for Genomics and Health over the last 10 odd years, and she's been the chair of GA4GH for the last few years. She's a very strong advocate and pioneer of open science and data sharing, really working to extend these approaches internationally. And today, fittingly, she's talking about tackling the VUS problem. So over to you, Heidi.
Great. Thanks, Catherine. It's a pleasure to start and end this series. So as Catherine said, I'm going to talk today about tackling the VUS problem, a problem that plagues us all. Let's see if I can get my slides shared again. Okay, so back to the slides. If you'd like a copy of these slides, you can scan this QR code or go to this link. I'll also show it at the end of the talk. With that, I'm going to dive into a little overview of the problem. This was a study we published a couple of years ago where we showed that the rate of inconclusive reports due to variants of uncertain significance was about 33% across 1.5 million tests. And of course, that rate gets higher as you make larger panels, like 76% for these big over 200 gene panels. And the percentage of variants in ClinVar, the highest percentage is variants of uncertain significance plus the conflicting. So this is clearly a big clinical problem that plagues us, plagues physicians, plagues patients, plagues researchers. So we're all sort of struggling for how to deal with this problem. So I'm going to talk today about a number of different aspects of this and ways that we're trying to tackle this challenge to make it less burdensome and figure out these variants. Now, we actually return VUSs to patients on a regular basis. Why do we do that? Well, it's an opportunity to build evidence immediately that many variants that we all return in the clinical testing arena or research testing, those variants are only seen often once in one family. And so you get one shot sometimes at interpreting that variant. So if you want to build evidence, do, for instance, parental testing to look for de novo occurrence, segregation, deeper phenotyping in that patient, other clinical follow-up. That's all got to be done in the auspices of returning that VUS so you can do those studies. Also, it might enable functional studies to be run and other research investigations, allows the individual, their physician, to track the variant over time as evidence may emerge in the future. Sometimes VUSs are used for clinical decision-making, hopefully not invasive things, but monitoring, things like that. in fact, physicians act on uncertain information in medical care all the time outside of genomics. It should be true that sometimes they make decisions within genomics when we're uncertain. Here's just an example of many of the types of evidence that are out there. This was a variant that we identified in an individual with hypertrophic cardiomyopathy who was actually a sperm donor. And we had great access to segregation data in this family. This is an example of the family themselves following up on their variant of uncertain significance, which was also in a gene of uncertain significance. And this is Matt Might, and he really started a campaign to identify other children like his, and ultimately that amassed a number of families and implicated that gene and disease. So there are positives to returning these variants of uncertain significance. Of course, where there are positives, there are negatives, and so there can be harms. Certainly it can create patient anxiety getting back these VUS, especially if they were not even expecting a genetic result, sometimes secondary findings and other things. Also, these can incur healthcare resources to manage or investigate a VUS, whether it's those follow-up clinical studies, gathering patient samples from family members, all sorts of work that can be done, but that incurs resources. There can be mismanagement. If that VUS is assumed to be pathogenic, which does happen in the clinical care arena, then there may be actions taken that really shouldn't have been with that level of uncertainty. And certainly if those procedures might be irreversible, surgical, pregnancy termination, et cetera. And we have even observed physicians using genetic testing less because of negative experiences, getting all these VUS back, and that can be challenging to their practices. There's even been lawsuits because of misinterpretation or lack of follow-up of VUS. This is a study or a case in the US that happened because a VUS was not followed up on with parental testing. Of course, these VUS getting returned to families, when they have no answers, they may, grab on to one of these VUS as the answer. This was a story of Esme Savoy, who had a series of different answers. First, a PCDH variant, 19 variant that was reported as pathogenic. That family started an entire online support group for raised money off around that gene, but then realized that her child was different than the others, and eventually that variant was ruled out. And they did exome, found another variant. She started another foundation for that gene. Then the exome was reinterpreted, and they found likely path and path variants in a different gene, a third diagnosis. So these journeys go on in many families. But if a third of all genetic tests have VUS, we can't possibly follow up on all of these VUS. And in fact, most, statistically speaking, will become benign. So it would really help to focus in on what might be real and what might be not in terms of a diagnosis. And can we do a better job of moving variants from VUS to either pathogenic or benign? That's really our goal here. And some of you may have seen some of the talk series from the bold predictions that came from NHGRI, the National Human Genome Research Institute in the US, around 10 bold predictions. One of those, of course, this was said now 5 years ago, was that the clinical relevance all genomic variants will be readily predictable, rendering the diagnostic designation of VUS obsolete. Wouldn't that be grand? So we're 5 years in, 5 years to go on this prediction. I don't quite think it's going to happen. That said, Doug and I wrote a follow-up piece from that talk and talked about, well, what would we have to do to actually try to realize that vision? Maybe not in 5 years, but hopefully not too far off. And we broke it down into 4 areas. One is improving the standards for variant classification. I'll talk about how that could have a beneficial impact. The other is sharing data, and we'll talk a bit about that. Also improving predictors of variant impact, using better training data sets, machine learning, gene and mechanism specific models, things that could improve what we have from what we have today. And then scaling the functional assessment of human variation. So can we make an assay for every gene and be able to test at scale all known or even theoretical variants that may not have been encountered to date? So I'm going to walk through those different areas, starting with the first one, as part of my community pillars of variant classification also, and that's the standards. So many of you know we've been working on a set of standards that I promised are coming soon, although I've said that before, but we have done one pilot. We're very close. We're just getting the calculator fixed so we can start the next pilot. So this is the group. And one thing I will point out, I'm not going to talk really about the new standards, but I will point out that one of the key things from my standpoint is turning it into a points-based system that's based on the Bayesian framework, and that for which the VUS can be subcategorized directly, because the number of points will directly implicate VUS low, VUS mid, or VUS high. And that will be hugely useful. I'll also note it'll be easier to get variants to likely benign, because honestly, in the absence of prior probabilities based on strong phenotype to genotype matching, most variants, statistically speaking, are benign. So we're making it easier to realize that truth. And here is just an example of a colleague in our hearing loss variant curation expert panel, Hila Aziz, who took 179 VUS, used a draft version of the framework. It's still been evolving a bit, but taking just a rough version of what we had and applying that to these 179 VUS, 49% actually became benign or likely benign. Another 55 went to VUS low, and we'll talk about what to do about that category in a bit. Some even moved up to likely pathogenic. So you can see already, just by these new standards, a movement of the VUS into other categories. So I think that's going to be helpful for patients. The other piece of this is the sharing of variant classifications in ClinVar. And I'm just going to give a couple of examples of where this really adds value. This is actually patient use of ClinVar. So here's a pregnant woman, 23 weeks. She had an abnormal ultrasound with an increased nuclear translucency. It's associated with both Down syndrome, but also brassopathy disorders like Noonan syndrome. So genetic testing had shown a VUS and a BRAF gene. Now, 2 clinical labs have already shared their data from testing with ClinVar, not the details, and that variant was submitted as VUS by both labs. And those entries were found by the pregnant mother. She reached out to the 2 labs. One was my own. We shared our aggregate data, and then we were able to reclassify that variant as likely benign and return that to the family, and the pregnancy was continued and they had a healthy baby boy. So it's just an example where sharing data can lead to good outcomes. But often the details needed to interpret variants are not always put in ClinVar. And I'm advocating for everybody to not just submit to ClinVar, but actually to include rich information. So this is a case where from our Rare Genomes Project, 50-year-old female with suspected limb-girdle muscular dystrophy, and we found one variant that was actually previously known to be pathogenic with a valid interpretation. We could not find a second variant. Ultimately, we found a 3' UTR variant. Of course, we all know variants out in this region. There's a lot of variation there. Most of them are not pathogenic, but this one seemed like it could, have an impact. And it was found it's segregated correctly with 3 of the affected children. So we thought, well, it's worth considering. Now we submitted this to ClinVar as VUS, which it certainly was, but we gave all of the details, the segregation data, the phenotype of the family it was found in, etc. And because of that detailed data, we ultimately got, and I will just stop and point out, there have been several papers published on what you can share in ClinVar without direct patient consent, and that is, higher level phenotypes like the overall diagnosis and individual variants. So please do go ahead and share this information. You can share more when they're consented, but you can share basic clinical data even when you have an unconsented patient sent for clinical testing. So anyway, back to the story. 2 other colleagues saw our entry in ClinVar and followed up with their own emails saying, gee, we've seen that same variant in a patient with similar phenotype. ultimately, we were able to aggregate 5 unrelated patients through this data sharing. These 2 colleagues hadn't entered that data in ClinVar, but because we had, it brought us all together, and then some additional functional analyses were done once we had 5 distinct families with this variant, and that ultimately led to implicating this variant as pathogenic with functional data. So really good example of the way we can use this data from all corners of the earth to really contribute to variant interpretation and solving patients' cases for families. We also do a lot of this work through our expert panels in ClinGen. This is just an example of a number of variants that were uncertain significance, sort of below this line here. All of these variants with the evidence from the public data was blue or uncertain significance. But when they aggregated additional data from all the labs that had submitted to ClinVar, they were able to move all 7 of these variants up to likely path or path. So again, we may all be saying they're uncertain, but when we bring our data together, we can then often classify these variants in one direction or the other. So really useful information, and we have expert panels across all of the different disease domains that are aggregating evidence and working with volunteer experts around the world. We have over 2,800 volunteers working in the ClinGen expert panels to share data, assess data, and classify genes and variants for their role in disease. And this is just a graph looking at the progress of ClinGen over the last decade that we've been working. The beginning, it was really developing the frameworks for our work, but then in the last 5 years, we've really started to scale the actual interpretation of genes and variants. Here showing the number of expert panels in variant and/or gene, as well as in green and blue, the actual number of genes and variants classified. So we're making progress, moving up. That said, with all this data sharing, there is still discordance, and those expert panels are working on trying to resolve these discordance. There's over 31,000 medically significant conflicting variants in ClinVar, and that is an area that we are working to tackle and working through some other strategies. So we've launched this new project that I call the ClinGen Curation of ClinVar project. Here, what we are trying to do, if you have a variant in ClinVar that has an aggregate classification, that's sort of the calculated classification from ClinVar, looking at all of the submitted individual submissions. So if they're distinct, like you can see in this case, some say likely benign, others say uncertain significance. So that is going to lead to a conflicting interpretation. So what if some of those were obviously wrong and they're leading to a conflict? So what we're doing is we're looking at those entries and seeing if there's obvious reasons that something may be incorrect or outdated. And so what we're typically looking for is at least 2 of the following qualities: old, an outlier, and lack of supporting evidence. One of those on its own is usually not sufficient. Just because it's old, it doesn't mean it's wrong. Just because it's an outlier doesn't mean it's wrong. Just because they didn't submit evidence, even though it annoys me, doesn't mean it's wrong. That said, usually when you have 2 of those together, it's more likely not correct. And so we will flag those entries. And what that means, you can see an example here of a flagged submission. We give the reason: outlier claim with insufficient supporting evidence. So let's say 10 labs said the variant's pathogenic, 1 lab says it's uncertain, but provides no rationale and no evidence for why they say that. We would flag that. Now, in this case, they get 60 days. So ClinVar sends out notifications to any labs that have flagged variants from our team, and they have 60 days to intervene. They could update the variant, add the evidence for why they're saying this, and that would reset, and then it wouldn't be flagged. But most of these, and we've done now thousands of them, most of these, there's no objection. They are, in fact, outdated and incorrect because knowledge evolves. So to date, we've done 14,000 records in terms of reviewing them in these conflicting variants, and we're able to flag 4,739, affecting 3,800 variants. And in fact, the conflict was resolved in most of those. 84% the conflict was resolved from flagging these variants. This is just a glimpse of when I'm in ClinVar and I'm looking at a record. I can click on a Chrome extension button in the browser, and this comes up. I can select immediately a particular record and then hit flag, put in the reason, add any comments, and submit it, and then it goes into our pipeline for going back to the lab. So this has been very useful to help resolve some of these conflicting variants in ClinVar when the information is just out of date. Here's an example. All those flag submissions are at the bottom of the record once they're flagged. But that said, we leave them in there because it at least tells you that particular lab saw the variant, and there may be useful evidence. Just because it's out of date doesn't mean it wasn't observed and there wasn't good evidence at the time. So, so this is just how we're leaving them in there in case anyone wants to or needs to contact those labs and or dig up evidence from the entries. If you have come across a variant that you want us to look at, you can submit a request. There's a little form on the ClinGen website. The URL is there, and you can just submit away and we'll look at it. Okay, the challenge with ClinVar, however, is there's lots of classifications now in there, millions of variants classified. So thank you to all in the community who are submitting to ClinVar. That said, there's often not sufficient evidence to use that information directly. So we really need to think about ways to more easily get access to that evidence and more less labor intensive, right? So I'm going to give some examples of trying to share the evidence base. One is our Nomad database. So this is just a challenging story. This was a story where it's very similar to the one where the families themselves went to ClinVar. This is a pregnant couple that had also an increased nuchal translucency, thought they had a RASopathy disorder, Noonan syndrome. And I had tried to contact the research group that had patient, ancestry specific data, and they would not allow me access to that data set. And that pregnancy was terminated as a result. And then later, when that data set was shared in aggregate, that variant was clearly too frequent to be pathogenic and it was benign. So that was a bad outcome because that population data was not shared. And all of you who work in the variant interpretation realm and genomic analysis know that the first step we do is get rid of all the common variants because they can't cause rare disease. So the more rich that data set is, the less work we all have to do and the less likely we'll misinterpret one of those common variants. And the reason we need global representation of this data is that a variant that may be absent or very rare in one population can be quite common in other populations around the globe. Like here, a third of individuals in South Africa have this variant that is absent in the US. So really important that we share this global population data. Yet a lot of this data is not representative of the global population, most of it being highly overrepresented in terms of European ancestry. Both our Nomad database, which is now much less representative with the UK Biobank data in there, and same is true of all the other data sets. So how can we work on fixing this and why do we need to? Well, if you look at the sample growth in our Nomad database going from version 2 to version 4, what you see is that there was a huge increase in data from European ancestry. And again, that goes back to the UK Biobank data set. But not a lot of increase in non-European, some, but not huge. If you look at the number of variants with an allele frequency over 0.1%, what you see is there's almost no change in that number for European ancestry, but a significant increase despite the small number of samples that came in for non-European ancestry. So what that says is we've largely saturated our understanding of common variation in European ancestries, but not begun to saturate it in non-European ancestry. So it's really helpful if we get this data. So we are working on two things. One, Nomad version 5, which we hope will be out before the end of the year, will include the All of Us data set, which is intentionally very diverse. Over half of the data comes from underrepresented minority races and 85% from underrepresented individuals in general. So that data will be very diverse, but even more diverse is through our project called Federated Nomad, And the goal here is that there are lots of data sets that are around the world that don't have permission to send them to us at the Broad where we process Nomad. But if they didn't have to share individual level data and we're just sharing aggregate allele frequencies, they could share that data. And so what we're doing is we meet monthly with all of these groups, teaching them how to process their data according to Nomad best practices, and then with the goal at the end that they would share their aggregate allele frequencies to combine them with our primary Nomad instance. So through catalyzation of the Global Alliance, which convenes stakeholders, we've been able to amass groups from around the world into this project, and now have all of these countries represented with promised aggregate allele frequency data sets from thousands, hundreds of thousands of individuals. And we're going to be representing this data. So soon, hopefully by the end of the year, one of these groups will have gotten their called variant and sent it to us, and we can start to merge this data and give the community access to it. So we're excited about that. Now on a slightly other area of sharing the evidence, this is now looking at case level data. So although I haven't talked about it in this talk, some of you may be aware of the platform called Matchmaker Exchange that we launched now 8 or 9 years ago, I think it's been. And the goal there was to create a federated rare disease database network where anyone with a candidate gene that hasn't yet been implicated in disease could put that candidate gene into this federated network. And through an API, we connected all the databases, and you could be matched with another individual researcher, clinical lab, et cetera, as the same candidate. It's been incredibly powerful at facilitating gene discovery for rare disease. However, you sometimes put your candidate in, you don't get a match, or you've got a variant of uncertain significance in a gene that's already implicated in disease, and you can't use Matchmaker Exchange for known genes. So what we'd like to do is take your variant and query all the world's genomic data, find it in one or a handful of patients, and get their phenotype back. So we call this variant level matching, and that is what we're working on right now. And just as one of the first examples of allowing your data to be queried by others, this is Variant Matcher, same group that brought you Gene Matcher, led by Nora Sabrera. And this is me logging in. Anybody can get access to this data set, enter your variant and your patient's phenotype, and you will get back the phenotype of those individuals you match with in that data set. And I was, in this case, half the patients were unaffected family members. The other half had all different phenotypes. So quickly I ruled this variant out, which is equally important as ruling a variant in terms of making progress on a case. So that is a very useful interface. We've been working in our Seeker platform that's used by thousands of groups around the world. There's over 70,000 rare disease cases in our Seeker platform, not counting the other instances of Seeker that have been set up independently. But we have consented all the researchers using our instance on Anvil to allow that data to be queried by others. And so this is just a view looking in a screen of Seeker where you're looking at a variant that was identified within this particular patient. And so I'm seeing the details of my patient, but below this line here, there can be one or more observations in other data in the system that is not yours, but owned by someone else. And we obscure the phenotype a little bit, but we show high-level areas of the phenotype. So one HPO term in genitourinary, one in musculoskeletal, and 2 in nervous system. So you get some sense of the phenotype. We give you sex and affected status, so a male, affected male here, quality metrics for the variant. And that information is usually enough to say, okay, this could be a match, or I could help rule this out. And if it's interesting, you can click the email button. It'll auto draft an email for you, include your patient's phenotype, and then you can send it off and ask for more detail on that case. This is now accessible within Seeker, and we've had matching going on for the last few months, and even had our own novel gene discovery through variant level matching. This was a non-syndromic retinal dystrophy gene that was implicated, and there were 4 different families, all had the exact same variant. So that's awesome, making progress with this platform. we are now connecting these data sets through a federated network. So you don't have to go to each one and search separately. And the first two that were connected were the Variant Matcher and Franklin. And you can see that when you're in the Franklin database, which you can publicly access and see the knowledge base in there, you can see for this variant in TSC1, the variant did not appear in Variant Matcher. And so you see that message right there, and there's no need to go to that database. It's telling you that it's not there. Or in this other variant down below, this variant was seen 726 times, 144 of which were homozygous. You can click on that link. There's then a link that sends you over to Variant Matcher. You can log in and get all the detailed phenotype. Or most likely with this level of information, you're probably not going to go there because that pretty much says it's not a rare variant for rare disease. But very useful because now you're seeing real time from one database, the connections to others. So we are working on connecting lots of databases through this. We have many groups that are joining through a monthly conference call, working to get more networks set up. We just moved the system to a new authentication mechanism. We've hooked up 3 of the data sets. We're waiting Franklin to move over to that new authentication system. But now you can see information through this federated network. So next steps, we're going to enable the delivery of phenotype data directly instead of making you log into a database for it, as well as expanding the sites that are supporting variant type queries. So not just do you have this variant, but do you have any loss of function variant in this gene types of queries. So lots to come from this network. So that's, one aspect is trying to share that underlying evidence so we can move some of these variants to path or benign through data sharing. But what else can we do to just simply reduce the negative impact of when we do report VUS and can't push them one direction or another? So another thing I'll go back to is this fundamental framework of VUS low, VUS mid, and VUS high, and making use of that in specific ways in the clinical lab arena. But I'm going to bring back some data from that paper I alluded to earlier, where we reported on the rates of VUS across all tests. So I showed you that rate increasing by the size of the panel. But now what I'm showing you is aggregating all multi-gene panels together and showing you that the rate of VUS in multi-gene panels at 33% roughly is statistically higher than the rate of VUS from exome and genome sequencing. Now, intuitively, that doesn't really make sense. There's more variants in an exome and genome, certainly ones we don't understand and can't interpret. So why would there be a lower rate of VUS in exome and genome sequencing? Now, keep in mind, this is those variants reported from exome and genome sequencing. It's not just what was identified in the case, but actually chosen to put on a clinical report and given back to the physician and the patient. And what we were able to show is that of the 2 labs that use these VUS subcategories already, the multi-gene panels, they report all VUS, often don't even get phenotype to contextualize those variants. And so the professional practices that for panel testing, physician ordered a specific panel, give them all the variants and the genes related to that phenotype, and they will make the decisions themselves. And so all these VUS low are given back. In exome and genome sequencing, you can't give back a variant without knowing the phenotype. You can't interpret an exome or a genome without the phenotype. So here you have geneticists making judgment calls about what variants are likely or probably or possibly relevant to that phenotype. And so the evidence level for these VUS low never meets a level of criteria of returning that data to a patient. And so you're seeing a reduction in VUS because of this clinical judgment that's going into play. So the other thing I'll point out, because we've now just published, oops, I didn't update this, it's no longer in press, it's published in Genetics and Medicine, apologies, but we published a paper on the rate of reclassification of variants that were classified in different categories. So what you're seeing here is if you take the VUS low category, what this shows is that of the variants that were reclassified, how many went down to benign or likely benign versus up to VUS likely path or path. And you see no red here. That means that no variants ever moved to likely path or path. And this is roughly over a 10 to 15 year period. In contrast, if you look at the VUS high category, almost half of those variants moved up to likely path and path or path. And in fact, they were the class of variants most likely to move at all ever. 10% of all these VUS highs did move up. And when they moved up, they went to path or likely path. So this shows the huge difference in these categories, subcategories of VUS. So with that, we're working on guidance to laboratories for reporting VUS just in general, as well as how to use these subclasses. And we just finished a draft. It's undergoing review through ACMG. But when it comes out, the key points that will be there is that labs should use these VUS subclasses, display them on genetic test reports, allowing clinicians and patients to focus on the most important VUS, and also exploring ways to de-emphasize or even not report VUS low. So maybe move them to a separate section or a supplement, or potentially even not put them on a report at all, which some labs are now doing. So I think that's going to have a beneficial impact on the negative impact VUS has had on our clinical community. So what else can we do to reduce the negative impact? Oh, sorry, this is a repeat of that slide. I moved the points to the other slide. With that, I'm going to end and just say we have started a clinical genomics laboratory community. I'm sure some of you are already participating in that. This is a collaboration between Global Alliance and ClinGen. And the goal is really to identify challenges and needs of our community, share approaches and develop solutions for these challenges, define best practices as a community, and then in collaboration with standards bodies that can put those best practices into formal policies. We meet monthly. We alternate 8 a.m. and 4 p.m. Eastern time so that different areas of the world can join at least half the time. And we've had 4 sessions. We actually had one today on June 16th. So our next one will be on July 14th, and we'll be talking about bake-offs to evaluate genomic analysis platforms. So I'll be sending out some information about that session shortly. With that, I'm going to just thank a large group of people that I have the pleasure of working with on lots of these projects, some of which I didn't have time to talk about today. As I mentioned, you're welcome to download a copy of my slides if there's anything in there you want. And with that, I'm hoping there'll be lots of questions. I saved lots of time so we could have good discussion and questions. And I'm going to stop sharing so I can see all your, at least some of your faces, Catherine's at least.
You can see mine. Thank you, Heidi, for a tour de force. And it's great to see the evolution of this over a number of years. And particularly, I was particularly struck by the strength of federating data, both at a gene and a variant level, which obviously has been a main focus of the Global Alliance over so many years. And I had 2 sort of more basic questions to start. And one is, do you find, I was just thinking that there is the strength of the end user is incredibly important, both in those being able to interpret results, but also those entering results. So do you find any issues in terms of people that are uploading variants in the variant representation? Is that uniform? Are the standards universal so that we're all talking the same language?
Are you talking about uploading to ClinVar or this variant level matching?
Yeah, uploading to ClinVar, for example, when it's querying a specific variant.
Yeah, so I would say that most everyone who's submitting variants at the 1-star status, which means you have to have used a classification system, be transparent with that classification. So in that category of at least 1 star, and the exception is OMIM and some other repositories are 0 star. But 1 star and above, they largely are using the current ACMG AMP 2015 professional standard for variant classification. That said, we all know that there's quite a lot of range of subjective interpretation when you apply this rule. That's one reason we've been working very hard for now several years to create version 4, which will have a lot of decision trees and a lot more concrete decision making when you're classifying variants, a lot more guidance, moving more to a quantitative points-based system. So while in theory, those are all done to a similar standard, in practice, there is quite a bit of variation and subjective and decision making. And so we're hopeful in the future that that will become more standardized. But one thing that I've observed over the last, it's been over 15 years, let's see, yeah, over 13 years maybe that ClinVar has been around. The movement towards standardization and commonality has been huge just by the transparency of seeing. I remember one lab started submitting ClinVar and all of a sudden they were clearly very different than other labs in their classifications. And then clinicians are like, why are you calling this pathogenic? And everybody else calls it uncertain. And that led labs to sort of recognize differences and change their practices and update some things. So I think just the mere fact of sharing and that transparency already moves things in directions. But I think over time we'll even get more consistency.
And at the other end of the spectrum, with genomics being much more mainstreamed, particularly over the last 5 to 8 years, I was remembering a 10-year conference in Texas, and I was a bit horrified that clinicians who were not geneticists were presenting VUS as if they were the answer, like as a diagnostic category rather than we don't know about this. How have you seen that problem increase, and how is that being approached?
So I mean, there's still a range out there, and there was sort of this view that, the labs doesn't want to take the liability of calling it pathogenic, but it really is. They just won't say it, that kind of attitude. And I think that was a big challenge when back in the day, you were often testing very few genes with a very high prior probability of positivity because sequencing was costly. So you didn't test the sequence the cystic fibrosis gene unless the patient had a diagnosis of cystic fibrosis. So chances were good that whatever you found was pathogenic. So I think that started this sort of notion that whatever you see is pathogenic. For better, for worse, as panels got bigger and bigger, and more use of genetic testing with lower prior probability, of even a genetic disease at all, let alone specific to a given gene. And as these panels got bigger, it wasn't just one variant of uncertainty, it was like lists of variants. And so all of a sudden you're a physician, you're like, well, I need one variant. They can't all be pathogenic. So maybe I should be more skeptical of this list of 7 VUS, right? So I think there has been, movement in the clinical, clinician community to recognize that, okay, a VUS really is a VUS. But that said, I think adding these annotations of VUS low, VUS mid, and VUS high is going to, like when we launched that 15 years ago in my lab, the physicians were so grateful that, because then they really could give a better sense of the likelihood that variant might be disease causing and really ignore and not follow up on the VUS low, that we're just going to create anxiety and be able to convey that to a patient to say, this is uncertain, but in this subcategory, we've never seen them move. So we don't think you should worry about that. That's really important for a patient.
And just one of the questions is, when will the new variant classification standards be released?
$1 million question. Well, I think we're hoping to launch the pilot within the next few weeks, if all goes well. But then there's that's take some time to do the second pilot. Then we're going to let everyone in the community join for a third pilot after we do a little bit of iteration. And then it's got to go through cycles of review by ACMG and the other professional organizations involved, allow time for public comment. So I think there'll be a final draft version, because we've been very transparent with the community. There are groups you actually might have seen, Franklin announce joining their pilot of our version. I did ask them to change their announcement to say draft version because it is still undergoing some change. but you're starting to see even platforms start to build tools. We're building a tool for the pilot. That's what's actually delaying it a bit, is finalizing this tool. It'll be a public tool from ClinGen. So anyway, long story short, the final published version is not going to be out until next year. I'm hoping the spring of next year, but we do hope that a final draft version will be released and being used by, we have over 100 labs that have signed up to test this. There are groups that have already moved to this draft version because they see it as much better than the current. So, so it's coming. I can't give a date.
Watch this space, but soon.
Watch.
Know that everyone is eager, which is great. Now, a couple of questions that have a similar theme, and like noting the manual review that occurs across thousands of variants. There's a couple of questions asking, do you foresee the development of more automated variant resolution and potential ways for AI to help solve the VUS problem?
So I think there are many ways that we can help scale variant classification. AI is one of them, and some of that is the aggregation of information from the public domain, where we have developed a tool called evidence aggregator that we've been using, that uses large language models to extract information and like cases and all the variants and evidence, even functional evidence, and bringing it together. So just speeding the aggregation of information. So that's one area. I think as these platforms get good, and our expert panels putting out thresholds for allele frequency that call a variant benign, these can be directly applied codes, better sort of interpretation of predicted loss of function with more caveats built in if the variant is beyond the NMD site, and really being able to better predict what might be happening with a splice site. Is it just shifting to another one, or is it actually disrupting and causing skipping? So I do think there'll be lots of information and ways that we'll be able to more quickly scale and incorporate evidence. That said, it's only the evidence that's accessible. And if all your data is locked up in your lab, all those cases with phenotypes, functional data, then no amount of AI or automation is going to extract that out of a siloed environment. So I think what's really going to change the game here is that data is made accessible and queryable through, APIs and other ways of gathering it. And that is what is going to allow us to really scale the interpretation of variation, is that data and evidence being shared.
And it's a great ad for the Global Alliance for Genomics and Health, which is about responsible sharing of data. So share your data, folks. Just a comment from Joseph Getz. Hi, Joseph. Thank you, Heidi. Very nice. Just a comment, Esme Savoy, as you mentioned her story. You may know her mum, Hilary, published a book about her genetic diagnostic experience, Around and Into the Unknown. It was us who convinced Hilary to take another test, as the PCDH19 variant didn't look like it. By now, Esme has a genetic diagnosis, a recurrent variant in a known disease gene. So thank you, Joseph, for that update.
Yes, thank you for that update.
So from MJ, Brian, what do you think needs to happen to move the dial with the sharing of the associated clinical and phenotypic information, which can have different challenges?
Yeah, I think, a lot of this comes down to labs, especially when the clinical labs aren't directly consenting patients. It puts them in a difficult position to really share that data in very open ways, right? There's only so much you can put in ClinVar without consent, although I think it's more than what most people put in today. But that said, we really want details. And I think what we need to do is create a mechanism to allow the lab. So this variant level matching, we've started with a few sites that are more on the research end or are using the public Franklin interface where people are putting data in there publicly that they can share. I think if we go to clinical labs now, we started building this platform and say, okay, I know you can't just dump your clinical lab, case level data into the public domain, but if you were to join a data sharing network where that information can be accessed through a authentication and registered access, because the nodes that are querying are known to the user. and you can monitor what's exchanging and ask people to agree to not try to identify anyone in that kind of safer space, I think it's going to be feasible to allow more data to be shared that hasn't been to date. And I think that's a bottleneck that the labs, when you call up the labs or send them an email, resources being a challenge, but a lot of them will reply and give very detailed information. And we all appreciate that, but it is a burden. And so I think the principle is there, but we need a system to make it easier for the labs to do it, to have less resources involved in sharing that information. So I'm hopeful that as we build out this network, that many clinical labs will partake, and that is a way that we'll get some of this data that's been siloed really out into the domain for use in interpreting variants.
Excellent. Now we've had a flood of questions, and I'm going to ask you a few quick last questions. We know there are some disease cohorts in NOMAD at the moment, which can sometimes make interpretation of the data in it harder. With a federated NOMAD, how will you deal with and minimize this impact?
Right. So for NOMAD, we exclude cohorts that we know were recruited for rare diseases, any rare disease. That said, data is coming in from biobanks. And so, anyone who signs up for a biobank, as a participant could have a rare disease. And in fact, most of NOMAD before we started putting biobanks was data from common disease case and control studies, diabetes cases, diabetes controls that don't have diabetes. That doesn't mean those individuals didn't have blindness or hearing loss or other things, right? So no data set is ever truly a control unless you spend a lot of resources making sure they're healthy, which no data set has ever done on all phenotypes. So, so we're with the Federated NOMAD project, we're asking the labs to follow the same protocol that we do, which is do not include cohorts recruited for rare disease,
Yeah.
but it's fine to include your population biobank data sets. And as this data set gets bigger and bigger, any small, anomalies, and we did actually find an anomaly. There was an increased number of CF Delta 508 alleles in NOMAD. We eventually tracked that down to the Mass General Brigham Biobank. There was a pulmonary clinic that was recruiting a lot of patients with cystic fibrosis in that one clinic. So there was a slightly higher increase in CF mutations as a result. But once that's combined with nearly 1 million samples in NOMAD, it pretty much washes out. So the larger we make this data set, the more a few little inconsistencies like that will get washed out. But I will say I heard not a rumor, but a truth that groups were eliminating the UK Biobank of fear that biobanks had rare disease patients. I actually would say the UK Biobank is the most healthy of all of the data sets out there because of the way that population was recruited. So don't do that. In fact, it's the Mass General Brigham Bank that's on the other side of NOMAD that actually had the cases we found. So yeah, it's a challenge. There's no perfect answer, but you just have to treat that as a population data set, not a control data set.
Question from Chris Barnett. Great talk. Thank you. Do you think VUS high should be reported in prenatal whole exome and whole genome sequencing? That's a tough one.
Yeah. So we are generally being very consistent in this recommendation that will be coming out that you only report VUS when they match a phenotype. And if you are doing testing that is in the absence of phenotype, like healthy screening, whether it be carrier, prediction in healthy individuals, you should not report any VUS. Whereas if you're doing diagnostic testing, then there's a phenotype and that variant is in a gene that matched the phenotype, then it should be returned. Now, we do talk about some exceptions. So if you are a clinician that has the resources to go into detailed consent processes and explain what is a VUS, what is a VUS high, all of this information, you're willing to go through that effort, and you've explicitly consented the patient to return all VUS high, sure. otherwise, so yes, a prenatal sample, if there's a phenotype, that variant could go back in the context of it explaining a phenotype found in a fetus. But if that was just a healthy, let's say a microarray done on a healthy baby, then the VUS would not be returned, even high.
And last question, I think, because we're running out of time, there's been a flood of questions. Heidi, can you comment on progress for non-SNV variation, e.g. SV repeat expansions?
Yes, so both in
RNA
nomad,
variation, actually. We're just a
yes,
new syndrome. I just
yes. So both in nomad, we have structural variants. Mike Tekalski's lab has led the calling of all the structural variants, and that is been done for nomad v4. It's also being done on v5 with the All of Us data set. There's also increasing interpretive resources from ClinGen. We have a dosage sensitivity work group that curates on both the dosage sensitivity of genes as well as chromosomal regions. So that curation is being done. There's also a fair bit of work on repeat expansions, tandem repeat expansions. We've released in nomad, and some people don't realize it's there, but there's a tandem repeat expansion data set for the population variation for tandem repeat expansions, as well as defining the pathogenic to benign range. Van Weisberg did that work in my team. It's there. It's being done on future data sets as well. And now he's expanding that to uncharacterized tandem repeat expansions, to develop resources to discover novel repeat expansions that could be implicated in disease. And that data is being generated both on rare disease data sets like the Greger data set. Ben has done that calling, as well as on population data sets like future versions of nomad. So this is a really important area of work, are structural variants and their roles in disease, and lots of work. And I think the use of long read sequencing, we're using that, others are using that, are resolving these structural variants better. Although I will say most of them can be detected by short read sequencing as well, even if they're not exactly resolved for the breakpoints.
So we're at time. Heidi, just a magnificent presentation. And when I reflect on what you've led and the amount you've done to change the face of genomics globally, it's an honor to know you, and I think you should be very proud of all you've accomplished. And just to think of the changes in the last 10 years is gobsmacking. So everyone, with that, this has been the last of our DNA dialogue. We're coming into the final weeks of 9.5 years of Australian genomics. This series has been hugely popular. We had around 280 attendees today. It's been hugely popular, commencing in April 2022 with Heidi. We could just feature Heidi on a monthly basis, I would suggest. But since that time, we've had 27 virtual seminars with over 3,200 attendees. So I'd like to thank especially Tiffany, Dorothy, Leanna, Will, and the Australian Genomics team who've been instrumental in delivering this series. So as we're winding up Australian Genomics on the 30th of June, I'd like to thank all of you who've participated. It's been a most wonderful collaboration, bringing together the genomics community around Australia and connecting it internationally. So it's been such a pleasure and a privilege to work with you all. But we're very excited about the establishment of Genomics Australia within the Australian Government Department of Health, that's going to build on these collective efforts. And I would say it's very rare that you start a grant from the NHMRC and it ends up as a government agency. So we should all be very proud of what we've achieved in terms of embedding genomic medicine within the health system. So thank you all. Have a great day. And again, thank you so much, Heidi.
It was my pleasure. Great.